Hello students, welcome back to my channel. Today I am starting a video which is the part 4 of the chapter Absorption by Roots. In this video basically I will be talking about a few experiments which is on the absorption and conduction of water in the plants. So without wasting time, let's get started with the video. So basically I will discuss about the 4 experiments which are very important from Icse perspective. Okay, so the first experiment that I will be talking about is the experiment to show that the roots absorb water. How will we do that? See, first and foremost, you can see there are two test tubes, test tube A and test tube B. Two test tube. Now the test tube B is the control setup and test tube A is the experimental setup. So what you have to do is you have to just place a young leafy plant. For example, the plant that has been shown here is the plant balsam. So you will just put that plant inside the test tube, and of course you should be very careful that the roots should be present in the plant. Okay, and you will put few drops of water in the test tube so as to prevent any loss of water by evaporation, so that evaporation will not occur. For that reason you are putting few drops of oil in both the test tube. You will do, but the difference is that in test tube B, as it's a control setup, so you will not place any plant in the test tube. Only in the test tube A you will put a plant there along with the drops of oil, but in test tube B there will be no plant. So what you will see in a day or two, if you leave this, then what you will see, you will find that the level of water in the test tube A, what has been? It will fall. See, this is the condition after some day. But there is no change in the level of the water in test tube B. Why? Because in test tube A there is a plant. So the roots of the plant are continuously absorbing water from the test tube A. For that reason there is a fall of water level in test tube A. But in test tube B there is no plant. So no one is absorbing water from that test tube. That's why the level of the water remains the same in test tube B. So from this experiment we can show that roots actually absorb water. Okay, now moving on to the second experiment. This experiment is to show that water is conducted upwards through the xylem. Okay, in upwards direction the water is conducting through xylem. This is the aim of the experiment. Now what we have taken, again we have taken the example of balsam plant, a medium sized young balsam plant. We have uprooted that. Then what we did, then we washed that plant. Then after washing we have placed that plant in the beaker which contain a stain. See the stain, pink solution, because it contain a red stain, eosin. And we should make sure that the roots of the plant should be completely submerged or completely immersed in the solution. This setup will be left for 3 to 4 hours. Okay, after 3 to 4 hours, when we again pull out the plant from the beaker, again we will wash it. Again we will wash the plant in tap water. And then what we will do, we will cut the transverse section through a blade. We will cut transverse section of root, of stem and of leaf. And from all the 3 transverse sections, when we see those transverse sections under microscope, we will see that the xylem vessel will appear distinctly red in color. See, distinctly it is red in color in all the 3 tissues, whether it's stem, root or leaf. So how it become red in color? Because the eosin stain is only present in this portion. That means roots absorb water. And how it is reaching the leaf? Because the tissue, the xylem, is actually conducting the water in the upward direction. That's why the leaf, the stem, the roots, everything, in each and every part, we can see that the xylem has become red in color, just because it has absorbed and it has conducted the red dyed water, red dyed solution. That's why. So that is the experiment. Now moving on to the third experiment. In third experiment, it shows that the conduction of water is actually happening through the xylem. Not any other tissue, but actually the xylem tissue is responsible for the conduction of water. So what we have done here, we have actually taken two setups, setup A, setup B. In setup A, we have removed phloem, only xylem is present. And in setup B, only phloem is present. We have removed xylem from the second part. So first setup, that is setup A, is showing a twig with phloem removed but xylem is intact. And setup B is showing a twig with xylem removed but phloem is intact. And both of the twig is immersed inside a beaker containing water. Now what will happen? After some time you will see that the leaves, in case of setup A, the leaves are fresh. They are rigid, they are strong. But in case of setup B, you can see the plant wilts, the leaves have wilted. Why? Because the leaves are not getting water, because phloem is responsible for the conduction of the food. Only xylem is responsible for the conduction of water. In setup A, where xylem is intact, there the leaves are getting water because xylem is conducting water. But in case of setup B, xylem is not present. So there is no conduction of water. As a result, due to the deficiency of water, the leaves wilt. This is the wilting of the leaves. This is also known as Godling experiment. Godling experiment. The ringing experiment is also known as Godling experiment. Okay, so from this we can actually conclude that xylem is the tissue which is responsible for the conduction of water, not phloem. Now the last experiment is to show that the food is actually conducted downwards through the phloem in the stem. Phloem is responsible for the downward conduction of food. How? Again we will perform Godling, ringing experiment. This time what we have done? This time we have removed the phloem from the stem. This is the stem. We have removed phloem from the stem. Xylem is intact. What will happen? A woody stem from where xylem is intact but phloem has been removed. After some time you can see bulging of the stem. See, I think you can see bulging of the stem, and this plant will die after some days. Why? Because photosynthesis is occurring in the leaf and the food is being accumulated at the end of the woody stem, because after that phloem has been removed and food or the starch is being produced by the process of photosynthesis in the plant. But since there is no phloem in the stem, so the food is not being conducted downwards till the roots. That's why there is bulging at the end of the stem where phloem is absent. So from here we can conclude that phloem is the tissue which is actually responsible for the conduction of food in the stem. So both phloem and xylem is important for the plant to live or to survive. Because if xylem is not present, then there will be no conduction of water. If the phloem is not present, there will be no conduction of food. So this is how 4 experiments are very important from this chapter. In ICC, one question you can expect from, or one experiment you can expect from this chapter, because this is very important. So I think all the 4 experiments are absolutely clear to you all, and that's it for this video. If you think my videos are informative, they are helpful, then please like and share my videos. And if you haven't yet subscribed to my channel, then please click the subscribe button and don't forget to click the bell icon. Otherwise, you will not get the notification whenever a new video will come in my channel. I will be back with a new video very soon. Till then, stay tuned to my channel. Thank you for watching the video.